Hello students, today we are going to discuss about the cultural characteristics of Klebsiella species. So mostly it will be Klebsiella pneumoniae I'll be talking here. So this Klebsiella pneumoniae colonies are large, shiny and dark pink in color. Two distinguishing characteristics you will see, that is lactose fermentation and the viscosity of the colonies. Then the encapsulated strains of Klebsiella species are also mucoid in appearance, which is a characteristic of the strains of this genus. Then there are various culture medias where we can use for the cultivation of this Klebsiella pneumonia in the laboratory, and most commonly used are like those of nutrient agar, MacConkey agar, medium, blood agar, etc. So where is the habitat? Mostly this Klebsiella is found, Klebsiella pneumonia. So mostly they are found in mouth, skin and intestinal tract of human beings. They are also found in soil, water and sewage. Then they are also found in bowel and respiratory tract of humans as well as animals. Then it is also found in hands of hospital personnel, especially those who are working in the hospital, like the ward boy or the nurses, all those people. Then they are responsible for community and hospital acquired infections. Then people requiring equipment such as catheters and ventilators are at high risk of this Klebsiella pneumonia. So we have the requirements, temperature, pH and all these things which we need to maintain. So what are the special requirements required? So in case of Klebsiella pneumonia, they has no complex nutritional requirements and readily grows in ordinary media like nutrient agar medium. Commonly, the nutrient agar medium and MacConkey agar medium is used for the cultivation of this organism. Then coming to the optimum temperature, this organism ranges from 15 to 40 degrees Celsius, but the optimum temperature for most of the strains is 37 degrees Celsius and usually cultivated at this temperature in the lab. Then the pH, we have the optimum pH. Klebsiella pneumonia can survive alkaline pH but not at acidic pH, and the maximum growth of this organism is observed at 7.0, that is neutral pH. Then also the pH requirement varies as per the strain of Klebsiella pneumonia. Then we also have another requirement, that is oxygen requirements. This organism, Klebsiella pneumonia, is an aerobic bacterium, that is, it grows best in the presence of oxygen, and it is also a facultative anaerobe organism, which means it can grow in the low oxygen environment. So these are the different requirements for this organism. So the first culture media where we are going to discuss is the blood agar medium. So how the colonies looks like in this media when we grow this organism in this media. So you can see the picture, right? In blood agar medium, the Klebsiella pneumonia colonies are non-hemolytic, because mostly in blood agar we will check for the hemolysis, right? So in case of Klebsiella pneumonia, you will see there is no hemolysis form. Then it can be considered as gamma hemolysis. Then the next media we have is the MacConkey agar medium. So out here you will see the colonies of Klebsiella. They are pink in color, right? Due to, what is the reason? It is due to the lactose fermentation. Okay, so which is of great importance in differentiating the Klebsiella pneumonia from other species or other bacteria present in the specimen, especially from gram-positive bacteria. Then the Salmonella species, which are also non-lactose fermentors, and the result they will produce is colorless. Then we have the next media, that is when we grow this organism in this media, what are the colonies we will observe, what are the characteristics observed. So this media, Cetrimide agar, you will see pigmented blue-green colonies, then the colonies exhibiting fluorescence at 250 nanometer and a blue-green pigmentation are considered as presumptive positive in case of Klebsiella species. So this is for the Cetrimide agar. Then we can also culture this media, this organism in liquid culture media like tryptic soy broth or nutrient broth. Out here, the growth of the bacterium will occur as turbidity in the broth medium, which is further analyzed for the morphology under the microscope. Then Gram reaction, we will perform the Gram stain. Then we will perform different biochemical tests and Klebsiella pneumoniae specific tests. So these are few medias which I mentioned where we can check the characteristics of Klebsiella species when we grow them in these medias. Thank you.